I just wanted to document an idea I had that has to do with paracatafungogenesis theory, which is the aspect of it which I call the gene gun. Uh, the gene gun presently is used to make recombinant, or was used to make recombinant DNA and structures and various insertions of different DNA into organisms that didn't have it. And basically, what it is a projectile that has DNA fragments that you desire, and you basically put them on a bullet, shoot the bullet into a plexiglass, have the plexi spray a target of cells. Those cells then ballistically get the fragments of DNA embedded in them. Usually, there's some sort of pellet size that is appropriate to make the delivery of this DNA. And what happens is you end up with a circle. Your sample has uh, a characteristic destructive inner circle, the circle where the forces are just adequate enough to get the delivery of the DNA into the cell, and then outside of that, it's the force is less, so that it doesn't do anything to any of the cells, and they probably survive as they were in the sample. But there's a circle that's changed, been modified by the insertion. Hopefully, they take it up and they begin to use it in the development of the proteins that you want the cell to do. So anyway, now in paracatafungogenesis, the idea that a parasite would take up residence in another organism, and then, let's say there were some sort of concussive event, i.e., a meteorite or some other explosive event that would happen, there would be a zone in which, just like in the ballistic scenario of the gene gun, that there would be a zone where any organism that may had a closer proximity to the genetics of another one was blasted into the nucleus of its host, so let's say, or vice versa, it could be, and then you would end up with a recombination of the that particular gene set, and the inner circle again would die because of exceeding the survivable concussive force. There would be a ring of life that would survive, and then outside of that, then there wouldn't be anything. You'd have all of this other thing. In a way, almost not, nothing to do with it, but it almost would be sort of like a fairy ring. But that's a different ring structure. Nothing to do with it. I'm just kibitzing in my own head. So anyway, that's the gene gun hypothesis, if you will. Um, Now I put that into a formulation of those things not occurring randomly. That that if you were terraforming or creating life on this planet, you could send those particular things uh, down like the Earth is a petri dish, the same way as the guy with the gene gun does to recombinate things. And then you get whatever organism that you think you're going to manufacture. So you can take your pick. I guess it still is open in both camps that if you wish to have it as intelligently directed versus randomly directed, you get to choose your path that you want to do. I particularly choose the intelligent idea. But I'm not going to kill you if you choose the other one, or I have no. So anyway, I just thought I'd document that. For some reason, I got it into my head to do like a little explanation of it. And the guy that developed the gene gun won a whole lot of awards and patents, and has, I believe, it was a professor in Columbia University, and he's since retired. And I also think that he may even be a believer in creation, and I could kind of understand how you might. Uh, I don't know if the the two were connected. The entire backstory. Anyway, enough of that. Thanks for watching, and